So last time we redefined the definition of evolution that we came up with at the start of the term to incorporate some population genetics terminology. So now we start talking about how mutation is basically based on a change in a mutation's frequency. That is, how many individuals in a population have a mutation over time. So if a mutation arises anew for the very first time, what happens to it? If it goes extinct, that is, if the individual that has that mutation doesn't produce offspring and pass that mutation on, that mutation goes extinct and there's no evolution. So it's only by a changing frequency that we observe evolution. That's why today we're talking a lot about mutation, almost entirely about mutations. So how does mutation occur in the first place? And then I told you a quick story about sticklebacks. I happened to discover this picture on my laptop, so I decided to throw it in even though we didn't talk about corn evolution last time. What was the take-home message about stickleback fish, the story from last class? Why did I decide to spend time talking about it? It was a solution to a problem that evolutionary biologists have had to face, a conceptual problem.
Whether it was one big evolution that changed, that turned them into what they are now, or was it a step? Was it multiple evolutions?
Okay, so to summarize your response, does evolution happen in great big leaps? One mutation causes a big change, or do several individual mutations, each of which make a small change, total up to major transitions in evolution, like the loss of stickleback body armor, the lateral plates, and their pelvic spines? And this was an example, and this is a summary from this image, that it didn't take multiple mutations, it didn't even take a single mutation when these fish colonized independently different freshwater habitats over the last 10,000 years or so, for all of those different populations of the same species to evolve the same trait, the loss of this defense armor. Because what these scientists found occurred was that the ancestral population, the marine population that colonized all of those freshwater lakes, already had that mutation or set of mutations that caused that phenotype. So here, there was a rare fish in that ancestral population that already had the useful phenotype. It was just at a very low frequency. There weren't many individuals in that ancestral marine population that had that trait. But when they found themselves in the correct environment, what happened? Or the environment where that trait was beneficial.
It became dominant?
Right, so then that variant, that phenotype became predominant. It became frequent. So that change increased in frequency over time in the populations, and that's evolution. That's why the freshwater populations look different today than they do in the marine environment. But it's not because parallel evolution happened. It's not that every one of these freshwater lakes had separate mutations happen that make the same trait. It's not convergent evolution, it's not parallel evolution. All of the mutations happened once, a long time ago in the past, out in the ocean, and that mutation was carried into freshwater habitats where it was selected for. But all evolution has to start with mutation at some point. Those new versions, alleles of genes, have to show up somehow. They have to appear for the very first time ever, like the stickleback loss of armored plates in the marine population. So the goals for today then are to explain how mutations occur, so be able to explain to me, maybe on an exam, processes that cause mutations. And then some review, hopefully, from genetics and other classes, explain the differing effects of different types of frameshift mutations, synonymous and non-synonymous mutations, and at the end, bring it all together to explain why this has anything to do with natural selection. So if we're going to study variation, one of the requirements for natural selection, we have to first talk about how variation shows up in the first place. That's mutation. Next chapter, right after the exam, we're going to start talking about population genetics. That's basically studying what happens after that mutation shows up in the first place. What happens to that mutation? Does it increase in frequency? Does it decrease in frequency? And those questions are dependent on a lot of things that we talk about in that chapter. So what happens to a mutation once it shows up has a lot to do with how many individuals are in the population, whether they're randomly mating with each other, whether or not that's a beneficial or detrimental mutation, hopefully, obviously. So that's how selected it is. Is it a really beneficial mutation? You'd expect that it's going to get more frequent in the population pretty quickly. Is it a bad mutation? Might kill the one individual that has it. It goes extinct within a generation. So that'll be what we're talking about after we finish mutation. That's where we're headed. So Darwin likes survival of the fittest. Before we get to that, we have to know arrival of the fittest. So how do these variants show up in the first place? And this is sort of a general categorization of the types of mutations that we could talk about. We've got point mutations, we've got insertion deletion mutations, major chromosomal rearrangements, and something that we don't often talk about as much, transposable elements, which are mutagenic. The pieces of DNA that move around in our genomes, jumping genes. Anybody have a good example of a jumping gene? From any class you've ever heard about?
I think they're corn.
Like corn. So Barb McClintock's work on, and we'll see that in a little bit. Thank you. Either looked ahead in the slides or you remember it from class, but either way, it's a good introduction to what we're about to see when we talk about transposable elements. Pieces of DNA that move around in our genomes and cause mutations. So maybe we should talk about skin color for a second. I have issues with ultraviolet light that some of you don't have. And that's because I don't have a lot of melanin in my skin that blocks my DNA from getting hit by high energy ultraviolet light. But what happens to your DNA when ultraviolet light hits it? Right, so ultraviolet light can cause two adjacent thymines, T's in our DNA, to literally covalently bond to each other in a way they're not supposed to. So that's shown up here at the top. So that's a thymine dimer. Doesn't look like the typical DNA structure. The two adjacent nucleotides are covalently bonded in a way that DNA polymerase can't replicate past. So that's what's shown here. So DNA polymerase gets stuck when it's replicating DNA and it hits this thymine dimer. Bless you. Yeah, it's that time of year. Those of you that are on Twitter saw my tweet a couple days ago about all the trees releasing their gametes into the air. It's good for them, I suppose, not good for us. So what do our cells do? What do my cells do when I go out and I get sunburned and all of my DNA is getting damaged in my epithelial cells? What does the cell do? What's its response to all this DNA damage? So one of the things is the cells could be programmed to undergo cell death, apoptosis. Right? If the cell's heavily mutated, maybe the best thing for the organism, not good for the cell, but maybe the best thing for the organism is to kill that cell so that it doesn't pass mutations on to daughter cells during cell division. Regarding mutation and how mutations arise, thymine dimers are mutagenic. It's not because the process of the covalent bonding of the two thymines together is mutagenic. It's because of what our cells do when they see thymine dimers in our DNA. So our cells have proteins that recognize, they go around scanning our DNA all the time for the presence of thymine dimers. And when one of those proteins senses a structure that looks kind of like this, two thymines that are bound together in an unnatural way, that protein turns on a new type of DNA polymerase. It activates a new DNA polymerase that's in the cell that can fix this problem. It can continue synthesizing the chromosome past that dinucleotide. All right, so that's Pol 3. So polymerase 3 can handle the thymine dimer. The problem with the thymine dimer is that our polymerase can't read that those are T's. So what does polymerase 3 do to solve this problem? It doesn't know what nucleotides are there. It's like we have two X's there. It doesn't know what the nucleotides are. So how is a polymerase to replicate your DNA when it doesn't know how to base pair, what to base pair?
Well, just put in anything because anything's better than nothing.
Exactly. So that polymerase just sticks in random nucleotides. So instead of putting in a G and instead of putting in an A and an A, for example, it has randomly incorporated a G and an A. There's a mutation. What should that first nucleotide have been? So originally, there was a T on the top strand, so there should be an A on the bottom strand. But instead, polymerase 3 put in the wrong nucleotide. It's great for the cell because now the cell gets to finish replicating its chromosomes and divide and survive. But our polymerase is designed essentially to make mutations in our DNA.
Doesn't polymerase 4 do that, not 3?
Oh, sorry.
Or 5?
Yes. My apologies. So it's polymerase 4 or 5 that come in to do that process, not 3. Thank you for correcting me. Absolutely right. Polymerase 3 is our typical polymerase that does most of the DNA replication. It's 4 or 5, and you don't need to remember the details of that. It's 4 and 5 that are the error-prone polymerases. They're enzymes that our cells have to replicate DNA, and they're supposed to make mistakes. Thank you very much, natural selection and evolution. You've evolved us enzymes that don't faithfully replicate our DNA, but they're there because they solve this one particular type of problem. So this is one way mutations occur. You get struck by ultraviolet light, your SOS, save our ship, I guess it should be SOC, save our cell, response turns on. That's these error-prone DNA polymerases come in and try to fix the damage by sticking in random nucleotides. Are there any vegetarians in the room? I'm mostly vegetarian. I eat meat on occasion, but does anybody really like a nice charred steak or hamburger, blackened tilapia? We've got at least one person in the classroom. Thanks for being honest. It doesn't start out being mutagenic. It starts out as this planar structure. The problem is, again, Mother Nature and natural selection has outwitted itself because we in our livers have these enzymes that are called CYP enzymes. CYP. Doesn't matter what it stands for. These enzymes are there to detoxify toxins when we ingest them or we're exposed to them in the air. So these are specific enzymes that are meant to take these sorts of chemicals and break them down so that we can excrete them from our bodies. For this chemical, though, for benzopyrene, what happens is those chemicals try to break it down, but this is as far as our enzymes that we have in our bodies can take the process. So this is the outcome. This is the output of liver trying to detoxify those benzopyrenes that we ingest. Supposedly most or some of you at least have taken organic chemistry. So what's the situation with this molecule?
Doesn't it make it highly reactive?
It's highly reactive because it's got an epoxide. If I could spell epoxide. EPOXIDE. Right? So that highly reactive epoxide molecule, when that molecule, there's this chemical benzopyrene that's a circular organic molecule. It's got lots of ring structures in it. It's planar. This is a chemical that gets generated through char broiling. It's in the black stuff that's on hamburgers, hot dogs, steaks, blackened tilapia, whatever. at very low trace amounts, but still, this is a mutagenic chemical. And it's mutagenic inside our double helix that epoxide can react with our base pairs, the nitrogenous bases that are the nucleotides. So this also causes mutations because, for very similar reasons to the thymine dimers, we're making covalently modified nucleotides that our normal DNA polymerases can't synthesize past.
Can it be carcinogenic, or is that just a problem?
So it is carcinogenic because it causes mutations. So basically, anything that causes mutations could be considered carcinogenic. And a lot of cancers rely on specific mutations in specific genes that predispose our cells to growing out of control, to make tumors, to be cancerous. So yeah, so basically anything that's mutagenic has the propensity to be carcinogenic. Hopefully this doesn't turn anybody off from eating the blackened steaks, but something we have to think about. Okay, so those are two ways that mutations are formed. We've talked about thymine dimers from ultraviolet light and benzopyrene. Both of these are covalent modifications to nucleotides that prevent typical DNA synthesis. Any questions at this point? Yeah, no more breakfast for you.
No, I was just thinking steak and eggs sound really good right now.
That does sound kind of good. Should we break for breakfast? Okay. So which type of mutation, missense, nonsense, synonymous, non-synonymous, is worst for us?
Nonsense mutation?
So a nonsense mutation, what's a nonsense mutation?
It creates a premature stop codon, which prevents the, I guess, protein to be actually fully translated.
Okay, so sometimes those point mutations can cause nonsense mutations, a change of a codon that produced an amino acid before, now encodes a stop codon. So at translation, the translation of the messenger RNA stops prematurely. So we've got synonymous mutations, those change the DNA sequence and do not change the amino acid that's encoded. And then they're the non-synonymous, ones that change the amino acid sequence. So these point mutations that can be caused by mutagens, UV light, et cetera, have numerous varying effects on the proteins. All right, so now it's your turn. Here are 5 different mutations. Each of these is supposed to represent an arrow. So you have these 5 codons that undergo different mutations, single nucleotide changes. So which of these 5, take about 2 or 3 minutes, which of these 5 is more likely to be acted on by natural selection? Feel free to talk to a neighbor if you want. We'll come back together in about 3 minutes. Right, that's the only codon that encodes tryptophan. So if you make any change to any of the 3 positions in the codon, you're definitely going to get a different amino acid. So is that synonymous or non-synonymous? Any of those changes would be non-synonymous. Any mutation to a tryptophan codon will change, I guarantee it, the amino acid that's encoded by that codon. So somebody pick a number between 1 and 3. 4? I heard 2. Okay, so then what nucleotide should we change position 2 to? A? Okay, so UAG. Oh, stop codon. Right, you can do that with any of the positions of tryptophan, change it to any other nucleotide, it'll change what's encoded there. So in this case, that's not only a non-synonymous mutation, it's a nonsense mutation. A nonsense mutation is what we call the creation of a stop codon. So where there was before an amino acid encoded, if a mutation makes that codon now a stop codon, that's a nonsense mutation. Otherwise, if a mutation changes one amino acid to a different one, that's both called non-synonymous and not nonsense, but missense. So where's an example of a missense mutation up here? Number 5. So what's UCG? Serine. What's AGC? Also serine. So that's a synonymous mutation. So you've made a mutation, but it didn't change the amino acid that was encoded. And in fact, so why did I put that example up there? What's similar about those two codons? How many mutations occurred to get from the left to the right? 3, right? No one of those nucleotides is the same, and yet it still encodes, so 3 mutations still encodes the same amino acid. Those are 3 synonymous mutations if they happen at the same time. What about CAA to UAA? Number 3. CAA glutamine Q to, UAA is stop. So that's another example of a nonsense mutation. Okay, AGA to CGA, that's arginine to arginine. Let's see. So we've got AGA to CGA, yep. So arginine to arginine, it's another, doesn't change the amino acid. So is it synonymous or non-synonymous? It's synonymous. It's a mutation, yes, but it doesn't change the amino acid. Will that be acted on by natural selection? If you have a mutation in your DNA, in a gene, but it doesn't change the amino acid that's put into the protein that's encoded by the gene, will natural selection act on that mutation? Is it beneficial if you have an arginine there before, or if you have an arginine in the protein there after? Does it cause a change in fitness? Probably not, because it's the same protein. It's exactly the same protein sequence, it's just a different DNA sequence. It encodes the same protein sequence. So when we're talking about which type of these mutations are more likely acted on by natural selection, what's the answer? Is it synonymous or non-synonymous mutations that are more likely to be selected for or against? Non-synonymous, ones that change the amino acid sequence of a protein. That's all about point mutations. One nucleotide changing to a different nucleotide. Then we've got frame shifts, which I know a lot of you, especially those of you that were with me last semester in genetics, have covered this a lot. So quick review. How will a frame shift, if you could summarize, how does a frame shift, shifting the reading frame, affect the protein sequence? Does it or does it not? Oh dear, it depends. Okay, so the effect of an insertion or a deletion of nucleotides depends a lot on context. So how many nucleotides were added or deleted, where in the messenger RNA, where in the gene does that mutation occur, etc. For a specific type of insertion-deletion mutation, the repeat expansion mutation, this is a mechanism. So I said that one of the learning outcomes today was to try to explain how mutations occur. We talked about 2 ways that point mutations might occur, thymine dimers, benzopyrene. this is a mechanism by which the normal act by our normal DNA polymerases of chromosomal replication can cause an increase or a decrease in the number of, in this case, repetitive DNA sequences. So if you have a part of your gene, which a lot of genes have these, that has the same 3 nucleotides or 2 nucleotides over and over again in a row, that's the repeat part of repeat expansion. This molecule of DNA, as it's base paired, sometimes temporarily becomes single-stranded. It melts, it denatures, just because that's the way DNA is. It's not always perfectly tightly base paired to the other strand. So if DNA is warm and its hydrogen bonds are releasing and forming and releasing and forming, you can get this mispairing when you have similar sequences all very close together on the DNA strand. And that allows for the addition, if you follow through the pathway, I'm not going to walk through it in detail, of one more unit of this triplet, of this nucleotide triplet. So if your two DNA strands don't pair properly, you get insertions or deletions of, in this case, units of 3 nucleotides. And this actually happens because this is the basis of DNA forensics. This is why you and I differ in the lengths of our genes, in many different genes across our genomes, and how you can take a DNA sample and PCR amplify it in a crime lab, presumably. Or in my lab across the street, if anybody wants to submit some DNA samples, I suppose we could do this. And many of us differ in the numbers of these triplet repeats that we have in our genes. And so you can tell 2 people apart based on how many CAGs there are in specific genes in our genome. And that's how forensic analysts do DNA fingerprinting. So I won't ask about details about this process. I just wanted to show you that there is a way that indels happen. This is one way, insertion, deletion, mutations. So again, time for you to do some work. Starting with this question. Can you formulate a rule for how insertion or deletion mutations will affect the protein? For example, I wrote one up here. One potential rule that you might come up with. Can you state, please, right now, on the spot, a rule that you think might be true for insertion and deletion mutations? So, for example, I've suggested here, if you reduce the number of nucleotides, does that necessarily reduce the length of the protein? If you increase the number of nucleotides, does that necessarily increase the length of the protein? Or are you going to fall back on that old standby, it depends?
Would it be too broad to say that insertions and deletions cause genetic variation?
So the question is, would it be too broad to say that insertions and deletions cause genetic variation? It's about as broad as you can get. It's unsatisfactory to me only in the intellectual sense for what I'm going to make us do next. But yes, you're absolutely right. That is the conclusion. That all that we conclude indels do is they cause genetic variation that natural selection may or may not be able to act on. And so now I want to walk through a little exercise with you to try to elucidate that, to try to point out why we can't make these sorts of rules. So your turn again. I've got two different copies of the exact same gene sequence up here. So on the top one, I'd like you, each one of you can work together if you want, to design either an addition of 1 or 2 nucleotides or a deletion of 1 or 2 nucleotides in each of these 2 sequences. On the top one, I want you to add or delete 1 or 2 nucleotides to cause that protein to become shorter. And on the bottom, I want you to do the exact same thing, add or delete 1 or 2 nucleotides that makes that same gene make a longer protein. So take about 5 minutes or so. Make a tiny little insertion or deletion mutation. Add 1 or 2 nucleotides, take 1 or 2 away to make the top one shorter, this one, and to make the same sequence, different mutation, to make the same sequence produce a longer protein. Suggestions then. It looks like everybody's pretty much done talking with your neighbors or working on this. So how can you make the top sequence make a shorter protein? What do we need to make a shorter protein? What determines the length of a protein? It's the distance between the start codon and the stop codon. So there's the start codon. If you want to make this protein shorter, how are you going to make a stop codon that occurs before the one that's here? So the current stop codon. How are we going to add or take away nucleotides to make a stop codon that shows up earlier? Bless you.
Can you try to like remove the start codon itself so the polymerase won't even be able to read it?
Okay, fair enough. That's cheating, but that works. So if you have a 3 nucleotide, although I asked for a 1 or 2 nucleotide deletion, I suppose you could delete the A and the T. Then there's no start codon, and then you make no protein. So is a 0 length protein shorter than, or I don't know. Now it's philosophical.
You can delete the two of the ends of the, because we want TAG, right?
So TAG would be a stop codon. So is there how can we make a TAG show up? So I suppose you could insert an A right there. That would make a TAG codon. So you'd have a methionine and then a stop codon. So you'd have the shortest protein possible for 1 amino acid. Does anybody come up with a different approach? Yeah.
You can insert a thymine at the, I think the glycine right there. So it makes you AA or T.
Okay, so you could insert a T. Let's see, we'd have to delete the C, insert a T to make a TAA stop codon. My idea was to delete this T, which means the next 3 nucleotides would be TGA, which is a stop codon. So there are lots of different ways, no one correct answer. There are lots of ways, lots of potential mutations that could be made by chance during evolution to create differences in protein lengths. And note that we've used both insertions and deletions. So there's no rule about insertions will always create stop codons, deletions will always create new stop codons. Both can do the same job. So that was 3 different ways to arrive at this solution. Hopefully the bottom one's easier. So what did people do to make the encoded protein longer? What sort of mutations might occur in nature when proteins are found to lengthen in certain species instead of others, as opposed to others? What's the easiest thing to do to make a protein longer? Pardon? So you can either add, so you want to add something? Let's see. What are we going to add? Do we add an A in there? Will that make this protein longer? That'll make it shorter because TGA is a stop codon. So then we'd be back at our first situation. We've got a 1 amino acid. So that's insertion, but it doesn't necessarily make the protein longer. It actually shortens it dramatically. So you can make an insertion. Yeah. Sure. Delete the T at the stop codon. Delete 1. Delete 2. Delete 3. Delete 4. right? Get rid of the stop codon. Guarantee that makes the protein longer. Right? And this is something that occurs frequently when scientists observe that protein lengths between different populations or different species change. Mutations have changed, either added a new stop codon, makes the protein shorter, or the mutation has occurred at the normal stop codon, changes the length, makes the protein that's encoded longer. We're just trying to explore how mutations cause change at the protein level, because proteins are how a lot of traits are formed. So we're going to explain how evolution happens and how traits change. It's going to be genetic changes that cause, usually, that cause protein changes. We'll skip this slide. The point of this was, if you were feeling a little bit more loquacious than you are currently, would have been to have shown that, and it's probably true, that all of you found both insertions and deletions could be used both to lengthen or to shorten a protein. So there is no way to predict whether a mutation that's an insertion or a deletion will have a specific effect to lengthen or to shorten. All four of those possibilities are true. Insertions can lengthen or shorten a protein. Deletions in the nucleotide sequence can either lengthen or shorten the encoded protein. Chromosomal rearrangements. Pass. That was only going to be a very brief treatment of that topic anyway. So transposable elements is something that I didn't talk about at all in genetics, and perhaps you've never heard about in a genetics class or an evolution class before. The point of this slide is to show that if you take the entire human genome and you start dividing it into its different components, how much of our genome encodes proteins, how much of our genome does not. There's quite a bit that's not DNA that encodes proteins. So that DNA that encodes proteins is this part. Roughly about 10% of the total DNA in our cells. So what does the other 90% do? If about 1/10 of our DNA is what actually encodes genes that make proteins, most of it's, junk DNA, or what's so-called junk DNA, especially repetitive DNA sequences. So something like 40% of our DNA is mobile DNA, like DNA viruses, things that exist only to make more copies of them and insert themselves into our genomes. Our genome's full of DNA litter. It's not that this litter is neutral, doesn't do anything to us. Sometimes it's bad, sometimes it's beneficial. It can cause evolution. All of these pieces of DNA in our genomes moving around. Maybe it's a term paper topic, for those of you that haven't started thinking about one. There's a great News and Views review article, a primer, to use if you want. Junk DNA is an evolutionary force, right? And it features Barbara McClintock's famous jumping genes, transposons, that cause the difference in corn kernel coloration, as many of you have seen in Biology 104, or will be seeing this term if you're co-enrolled. Right. This is insertions of movable DNA when they stick themselves into the corn kernel color gene. It changes the corn kernel color from yellow to purple, or from purple back to yellow. And that's why you see these sorts of maize cobs. And it's all based on, this is evidence of, and this is what Barbara McClintock proved a long time ago, this is DNA moving around in the genomes of these cells that causes these color changes. This is evolution in action. It's evolution happening before our eyes. And this is just a tally in terms of percentage, different types of mutations, very similar to the first table or graph I showed you. What percent of our genome is made up of, and the different chromosomes are made up of these mobile DNA sequences. So something like 50% of all of our chromosomes are this category of DNA. DNA that does not encode proteins that our cells use. Half. What a waste of energy. You have to replicate all of that DNA every time a cell wants to divide. But sometimes those DNA sequences do good things for us, and some of the times they do bad things. So brief summary, arrival of the fittest. We've looked at a few different types of mutations. We talked a little bit about benzopyrene, thymine dimers, how insertion and deletion mutations can occur, replication slippage in the triplet repeat expansion example. We talked about different types of mutations and whether or not they're visible to natural selection. And we touched briefly on transposons. The question I want to leave you with is this bottom one. This is really critical. This is a cornerstone of evolutionary thought. Do mutations occur randomly? What do you think?
Some yes, some no.
Let's pick. Oh, you guys are so good. You know the answer to every question. It depends. Don't try that on an exam. Unless you back it up with facts. So it depends. So not every type of mutation is random. But in general, what do you think evolutionary biologists would want to argue? Do you think we try to argue that mutations are random or non-random?
Random.
Yes. This one that was the point. You can't play both sides on this question. You have to come down on one side or the other. The theory of evolution is based on the concept of random mutation. That any mutation is possible, that evolution doesn't have foresight, that there aren't specific mutations that occur because they're beneficial. That's the totally backwards way of thinking about evolution according to evolutionary biologists. That random mutations occur, and the ones that are beneficial get selected for, and the ones that aren't get selected against. So a lot of evolution is trying to prove, a huge field in evolution, is trying to demonstrate whether or not mutations do occur randomly, or if there's some bias to the types of mutations and the locations that mutations occur in. And we'll talk about this a little bit next time. Right now I'm sending you a PDF. It's got instructions in it. We don't have much time to work on it now, but if you open it up, you'll see that there's a graph. This PDF has both a plot that I'd like you to fill in, as well as instructions at the top. Here's the brief details. So assume that this is a question on the exam we're going to have in a little over a week. This is why I'm doing it this way, because this is exactly like what the exam is going to be. It's going to come to you on a Google Classroom PDF. You're going to get it on your tablet. You're going to have to draw something on the PDF and send it back to me on Google Classroom. So this is your first, we've done this before, but this is the first real practice for what it's going to be like a week from Friday. So open this PDF in an app that you can use to annotate the PDF, draw on it. Follow the instructions that are in the PDF, the text instructions, and then attach this drawn on, your version of the annotated PDF, back to this Google Classroom assignment. That's the assignment. Turn it in. It's due the end of today, so 11:59 p.m. and 59 seconds. You don't have to do it right now, which is good because we're almost out of time, but pretend that this is an exam. Your goal is to have this back to me attached to Google Classroom by midnight. If you don't succeed in that, you've got time. It's not really due at midnight. Pretend it is. You've got time to go get help at the Discovery Hub or from me at office hours. We've got until next Friday to figure out how to do this process on your tablet in class. Because next Friday will be 8:50 in the morning. It better be attached to Google Classroom to me, because that's the end of the exam. So this is practice. What's going to happen is I'm going to give you feedback on how well you succeeded at doing this process with this assignment. Next Monday, I'm actually going to give a mock exam. So we'll do exactly the same thing in class on Monday. The goal then will be to practice doing it by 8:50 in the morning. So you got 2 chances to practice this before the exam. This one and one on Monday. So do this. It's important so that you don't get stressed out next Friday.